Good morning, everyone. I'm delighted to participate in this workshop and to share some of our knowledge and things, things that we've learned and research on the genetic management of small populations. I'm a population geneticist by background and conservation geneticist by passion, I suppose. And I've been working on endangered mammals and birds across New Zealand and Australia for a long time now. And I'm particularly interested in how small populations change in response to human activities, especially conservation interventions. So that's things like if you bring animals into a zoo population, how does that population change? How can we maximize its resilience? Or if we translocate animals to offshore islands, if we do other kinds of translocations, genetic rescue, all those sorts of things. That's my interest.
And yeah, hi, I'm Kate Farquharson. I'm a postdoc in the Australasian Wildlife Genomics Group at the University of Sydney. I completed my PhD in 2020 with the wonderful Catherine as my supervisor. And so my PhD was on genetic management of captive breeding programs. And since then, I've continued with my postdoc work looking at how to integrate genetic information into management and also moving to some of the genomic information, integrating SNPs and whole genome data as well. So yeah, thanks, Catherine, take it away.
Thanks very much. All right, so we'll get started. The format for this session this morning will be, I've got a few slides here about genetic management of small populations. I'm going to talk through some of the genetic issues that apply in small populations, some of the genetic processes that we're most interested in from a conservation perspective, and then talk through a few examples of the different kinds of management actions that are available to us when we're trying to preserve the genetic resilience of populations. And then after that, we'll go into a practical session with it Kate's going to lead, where we're going to look at the analysis of some real data from Tasmanian devil and look at how we can quantify some of the genetic processes that are affecting that species. Before we get started, I'd like to recognise and pay my respect to the elders and communities of the lands that I'm currently on. I'm currently at the University of Sydney. I'm on the Gadigal land, and I acknowledge the traditional owners of the lands that you are all on. I know many of you are down south on the sunny coast, and there are lots of people on Zoom, so we have a lot of people from all over the place joining us today for this workshop, and I acknowledge thousands of years of knowledge that have been exchanged on these lands for the benefit of our beautiful natural environments and for all the people who live here. I wanted to start by putting up this slide, which gives us a little bit of context about the hierarchical nature of genetic data when we're thinking about species and biodiversity in particular. So this illustration shows some of the levels of genetic information and genetic diversity that we can think about when we're looking at wildlife species or natural populations. So at the highest level, we've got genetic differentiation, which may be differences between species, but it could also be even within a species, differences between different populations and different groups of organisms that are living in different areas. Then if we go down to the next level and we zoom into an individual population, now we can start to think about diversity of that population in terms of richness and heterozygosity. We can think about the inbreeding of that population, effective population size, and the movement of individuals between populations. We can then move down to an even finer grained level of thinking about genetic diversity and look at it at the individual level, where we're looking at the diversity of individuals, which is essentially their heterozygosity, maybe their inbreeding level, and then how that diversity is distributed among individuals in that population via the mating system. So there are lots of different sort of ways that we can think about the genetic diversity of a species. And at all of these levels, there are important evolutionary processes that impact the potential of that population, evolutionary potential, or the genetic resilience, as I've been saying. So that's the ability of that population to adapt. How all of these different variables change across space and time is really useful for conservation management. So I just wanted to remind everyone that information at all of these levels can inform a range of different management strategies. So let's think about what happens when species decline. And a lot of species are declining. We all know this. This figure on the side is from the Living Planet Index. We know from this data is from 31,000 populations representing over 5,000 species, showing a decline in the diversity of those populations of 69%. So we know this is a big issue and something that we need to be thinking about. So when species decline, thinking from a genetic management point of view, small population size leads to a variety of effects, but the main things that we're thinking about here are genetic drift, which is an increase in the genetic stochasticity of the population, inbreeding, which is an increase in the mating among relatives, and also population fragmentation, which exacerbates both of those other two effects. So the question then becomes, how can we measure and manage these processes? And in the next few slides, I'm going to go through each of these processes in turn and think a little bit more about how that looks from a conservation perspective. So conservation genetics of small populations. First, let's think about genetic drift. And if you've interacted with conservation genetics before, you might have seen a figure, something like what I've got here on the side. These are simulated data that show the change in the frequency of an allele that starts at 0.5 and then how that frequency changes over 50 generations. And each of the colored lines is a population. And in the top panel, there is a finite population size of 50, and in the bottom panel, a finite population size of 500. So you can see immediately that with the smaller population, the one on top, that there's a lot of jagged lines. The lines are bouncing around, and that's just the changes in allele frequencies due to chance. And some of those lines, you see that they go all the way to the very top. Near one, there's a light yellow line that does that towards the 45th generation. And you look down the bottom, you can see there's a gray and a kind of tealy colored line. Both of them end up going all the way down to 0. So in those cases, that allele has gone to fixation in the case of going to 1, or it has been lost in the case of going to 0. And that's simply chance. There is no selection happening in this population, in these populations. Whereas, of course, on the larger population size, you can see none of the lines really even get close to going to 1 or 0. And you could extrapolate that in even bigger population size, that those jagged lines, we would see much less wiggling around that original allele frequency. So the population size really matters. And that is those jagged lines that you see, that is the stochastic effect of genetic drift. So to put that into some words for you, we know that genetic variation underpins phenotypic variation and therefore the adaptive potential of populations. That's why we're worried about genetic variation in general. Genetic drift is this stochastic change in allele frequencies from one generation to the next. It has a much stronger impact on small populations than on larger ones. Excuse me. In very small populations, genetic drift is a stronger force than selection for driving genetic change. And we can see that in that simulated illustration on the right. So what that means is that the allele frequency change from one generation to the next is driven more by chance than by selection. So selection operating on harmful variants, trying to remove harmful variants, may no longer be effective. Those harmful variants may increase in frequency or may even become fixed just by chance. And beneficial variants, perhaps things that are at a high frequency due to selection, if the population were larger, if the drift is now stronger, those beneficial variants may end up being lost. Now, the longer a population stays small, the longer genetic drift will impact the population diversity. So what that means is populations that remain small for a long time will suffer greater diversity losses than populations that perhaps undergo maybe a modest decline or they increase in frequency, increase in size, I should say, very quickly. So if a population goes along, gets small, and then stays small, that is a lot worse than a population that gets small and then gets bigger again. Obviously, a population that stays big is the best case scenario. Next, I'd like to talk a little bit about inbreeding. So inbreeding has multiple definitions in population and evolutionary genetics, but here I'm going to focus on inbreeding among close relatives. So that's when a population is small and individuals end up breeding with their close relatives, even by chance. So even if the mating is random, they may still end up breeding with a very close relative. You can imagine in a very large population, if individuals are breeding at random, the chance of mating with a very close relative is very small because there's just lots of individuals to choose from. But in a small population, that's not the case. There's just a very high chance that you'll breed with a relative. Now, this kind of inbreeding, it can accumulate over generations, and it increases the expression of harmful recessive alleles. So what that means is when every individual in the population is carrying some mutant alleles that are typically recessive, but maybe harmful. But if you inherit that allele from close relatives into a single individual, then those alleles can become homozygous and then they become expressed. So as I said, inbreeding is inevitable in small populations simply because there's just not that much choice in who to breed with. Inbreeding reduces individual survival and reproductive fitness. That's because of those harmful recessive alleles. We call that inbreeding depression. It is a decrease in fitness. Fitness is depressed. And ultimately, if survival and reproductive fitness are reduced, then that can risk population loss. And this effect of inbreeding being harmful, this reduced fitness, is a widespread observation for normally outbreeding diploid species. So inbreeding depression is a very common outcome of inbreeding, something we need to be mindful of in small populations. So the third process is fragmentation, as you can see in this picture, where you've got small patches of habitat or green space that have been modified by human activities so that it becomes difficult for individuals to cross those modified landscapes and to breed with one another. So land use change modifies the landscape in lots of different ways. It doesn't have to be as stark as this image I have on the slide. It could be more subtle than this. But essentially what it means is that habitat patches now exist in a mosaic of inhospitable environment. And individuals then may be unable to cross that inhospitable environment, and movement across the landscape is either reduced or prevented entirely. So population fragments that remain, therefore, may be small. So even if there are many population fragments, individual fragments may be small, and therefore drift and inbreeding, which I've already talked about, become big issues for those little population fragments. What we see is that a fragmented population is generally at greater risk than a contiguous population of the same total size. It doesn't always apply, but that's often the observation. The reason for that is because individual population fragments are at greater risk of being lost. So if those fragments get lost, then we're losing total diversity. We can use genetics to characterize population fragmentation and support conservation planning. There's a couple of ways we can do that. We can use genetic data to try and infer whether populations truly are fragmented or whether there's a small amount of migration between fragments, so they may indeed be connected. Or we can use that information, if we have identified isolated fragments, to rank populations in a risk assessment framework and identify which fragments are likely to need support and which fragments may be doing a bit better, and we could use those as source of individuals to support those other populations. So this, of course, is in combination with ecological data, biological data, things we know about the species and how they move in the landscape. Genetics is just another tool that can help support these kinds of inferences. So how do we measure some of these population genetic processes? I thought it might be useful to introduce this concept of essential biodiversity variables. This is a concept that is used in a variety of different biological fields of study. And the idea here is to identify a set of metrics or a set of statistics, a set of measurements that help us to, as it says here on the slide, aggregate, harmonize, and interpret biodiversity observation data from diverse sources. So as I said, from diverse sources, there's lots of different ways that we can measure genetic diversity, lots of different tools, lots of analysis techniques. I know that you've been in this workshop working with some very cool analysis techniques for looking at DART data. If you've collected a fantastic DART analysis data set, you might want to compare that with someone else who's collected data in a different way. So we need to be able to speak a common language and make comparisons across those data sets. Or of course, if you want to compare your data with perhaps archival data or historical data. The second objective of essential biodiversity variables is to use these metrics as inputs and validation to forecast status and trends of biodiversity and to support policy and decision making. So my colleagues and I had a lot of discussion about what kinds of metrics would be useful for measuring population genetic processes. If you want to learn more about that process and about the rationale for the different metrics that we came up with, you can have a look at our paper. I've got the link on the slide. And I should say, I've got links on all the slides for more information about the different processes that I'm talking about. But essentially what we came down to is that for measuring genetic change in populations, the key variables here, or the key metrics, are genetic diversity, genetic differentiation, inbreeding, and effective population size. So just a little bit more about these particular metrics. Genetic diversity can be measured in a couple of ways. It can be measured as the richness and evenness of a population. So that could be the number of alleles, it could be expected heterozygosity. Genetic differentiation could be the number of genetic units and the degree of differentiation among them. It could also intersect with the concept of the number of evolutionary significant units. And you may also have heard the term the genetic management units. There's a few different ways that we can count and compare units of genetic diversity in a species. In relation to inbreeding, we're largely thinking about the degree of relatedness between pairs of individuals, co-ancestry. So that's, as I said, that's mating between relatives, or identity by descent, which nowadays with genomic data, we can get to measure some really cool statistics around identity by descent. And effective population size, which is the size of, is a sort of theoretical value, which I think you're going to hear more about in other sessions in this workshop, so I won't go into in too much detail, but the size of an ideal population that loses genetic variation at the same rate as the focal population. So why this combination of four metrics? I'm sure that many of you can think of other kinds of statistics that could be useful for monitoring the genetic status of a population. And I should say this is for population genetic diversity as well. So we're not talking here about things like phylogenetic diversity and differences between species. So we're looking at within a species here. So why this combination? For genetic diversity, if we see a decline in genetic diversity, then that will indicate a loss of adaptive capacity and or perhaps increased levels of inbreeding. If we see a change in genetic differentiation, then it may mean that we're losing independent evolutionary or demographic units of a species, and increased differentiation may indicate more fragmentation or reduced gene flow between units. It can also potentially be an indicator of genetic drift as populations are drifting apart from each other. Inbreeding, as I said before on my earlier slide, increases the chance of the expression of deleterious harmful alleles, which reduces fitness. And effective population size is important because lower NE increase is associated with increased genetic drift, increased inbreeding, and the future loss of genetic variation. So to put it another way, here's a beautiful illustration from our paper, which you can have a look at later if you're interested. And in here, we've sort of got our 4 main categories of genetic variation and a few different ways of thinking about them. And you can see in the green shaded background, that's sort of our favorable condition where richness, the number of unique variants in a population is high, heterozygosity is high. For genetic differentiation, the number of genetic units and the distance between them, you can see both high and low are shaded because it can be a little bit complicated. You need to think about what is the driver of that differentiation, whether it's evolutionary processes like local adaptation or whether the drivers may be genetic drift and human activities. And for inbreeding, of course, we want low inbreeding, low relatedness among individuals, and effective population size, we want that to be high. So here's an example of a study here in Australia that my colleagues and I worked on looking at calculating EBVs in a local context. And that project was looking at a reintroduction of bush rats on North Head. Bush rats are a native species here in Australia. This research was led by my honours student, Aidan Wright, and in collaboration with Australian Wildlife Conservancy, and my colleague here at University of Sydney, Tom Newsom. So in this species, what we looked at was the introduction of these animals to North Head. So here on this map, you can see a zoomed in picture. You can see Sydney is sort of represented by all this white and grey coloration on that panel B, you can see there. So North Head is very much in Sydney, and the source location is some parks just north of Sydney. So we had 4 different trapping locations that we used to establish the population on North Head, and they were separated by a major road as illustrated by the big black line in C. Now, the population was established over a series of translocation events. So initially in 2014, there was a reintroduction where 73 individuals were released, and then there were 2 follow-up supplementations in the years following. And because these individuals came from a range of different localities, some of the management questions that AWC had for us were in relation to whether that mixing of source populations had successfully increased the population diversity, or at least given it a high diversity to start with, and then over time, whether that diversity was being maintained. So that's where we came in, and we collected some SNP data using DART sequencing and had a look at the population. So the details of the outcomes of this translocation and the diversity that we saw are not super important here for our conversation. If you would like to learn more, please check out our paper. But I wanted to illustrate that we were able to evaluate all the EBVs, a whole variety of genetic metrics for this population. So we looked at genetic diversity of the source populations and also the resulting population at North Head. We looked at genetic differentiation among the source populations and then among animals at North Head over time to see how the population might be changing. We also calculated processes related to inbreeding by looking at within population kinship, so how related all the individuals were to each other, how that might have changed over time. Because all our SNPs were mapped to reference genome, we were able to have a look at just in general genomic SNPs, but then also look at SNPs that fell within exons. So we had some interesting questions around whether there was any functional changes. And then finally, we were also able to estimate the effective population size of the 2 source populations and see whether that changed over time as well. So when we think about the genetic management of populations, let's take a little step back and think about in general, what is our objective? What are we trying to do? I mean, of course, we're trying to prevent extinction, but how can we use genetic data to do that? In the main, what we're trying to do is maintain the adaptive potential of a species and its populations. And for the most part, that means maintaining the genetic diversity of that population. We're also trying to maintain individual and population fitness, because that supports the first goal. So if individuals and populations are demographically stable or growing, then that will help to increase the population size, maintain the adaptive potential. And essentially this is involved with reducing the effects of genetic drift and inbreeding via increasing the effective population size. So you can see that these three objectives, they're not completely independent, and trying to improve one of these objectives will probably help improve the other ones as well. They're all interrelated objectives for trying to prevent extinction. So some of the questions that you might ask yourself in a genetic management context are, first of all, is genetic management necessary? Is there evidence that this population is at a high risk of losing diversity via drift, or a high risk of increased inbreeding? How big do these populations need to be self-sustaining? For that, you might need information about whether population fragments are connected to one another. What is the relevant timescale for monitoring? To a large degree, genetic change happens over generational time, but also over years. So we need to know a little bit about biology and mating system of the species. And then when we weigh up our options, how much loss is too much? So we're trying to balance the genetic objectives with all kinds of other objectives for managing a species to prevent extinction, and then also managing the costs and coming up with a large sort of decision tree risk assessment framework. So managing genetic diversity, even once you have identified that there's a problem, what are you going to do about it? So we have a variety of options for action. As I said, in the main, the objectives are to either increase the population size, or I should say increase its effective size, and or to increase connectivity among population fragments. And there's a few ways you can do that. You could do a variety of ecological interventions to remove or decrease threats that improve demographic parameters. You could increase or improve habitat, so potentially removing predators from an area, or restoring a patch of habitat where individuals can live. We can also increase connectivity among populations. That may involve either naturally increasing connectivity by building habitat corridors, or human-mediated movement, which is translocations. Or we could use captive facilities or a breeding program to support population recovery, to increase the population growth rate, improve demographic parameters, that kind of thing. And there's other things, of course, that we can do. These are just some examples. So the first step in this process, and where your genetic data come in, is in the monitoring to support planning. So the first step is to really understand the situation we're dealing with. First of all, we might want to quantify and predict what might happen if we do nothing. So that gives us a null hypothesis. It gives us a baseline to start from. What if we just leave this population as it is? How can we use our genetic principles, genetic monitoring, and genetic knowledge to think about what will happen if we just do nothing? If we're not comfortable with what will happen if we do nothing, if it looks like populations will decline or the situation will get worse if we do nothing, then the next thing to do is to rank populations in terms of their genetic vulnerability. If you're in a fortunate position with a species that has multiple populations, then identifying which populations are doing worse can be really useful for targeting your management. Obviously, that doesn't apply to all species. Some species, there may only be one population remaining. In the process of ranking populations, then you might identify relatively healthy populations. So there could be a useful strategy around trying to connect healthier, more genetically robust populations with other populations that have lost diversity, so that the healthier population can support the population that's dwindling. And perhaps even answer some specific management questions. So these are some general principles, but some specific questions might be, would translocations help achieve genetic management goals for the species? When do translocations need to occur? How many individuals should be translocated? Where should we get them from? Where should we put them? And maybe even which individuals should be translocated. And to some degree, ecological and biological data can, of course, really help with some of these questions. For example, which individuals should be translocated, knowing a little bit about the mating system of the species and knowing about its natural dispersal tendencies can help to increase success there. So here is an illustration that shows some of the steps involved with planning a major genetic monitoring program. The important point to take away, well, that I would like you to take away from here is that there are a lot of steps that involve a variety of options. So there are options and there are different ways to achieve similar outcomes. And also, as we look down the bottom of the figure, you can see that there are a variety of different steps involved with sharing and communicating the results of the outcome, the results of your activity. So by doing that, we can learn from each other and learn from experiences about the best ways to manage species. So if we do a management action, maybe we do a translocation, maybe we restore some habitat, hopefully in the hope that the population will grow. Maybe we bring some individuals into a breeding program so that we can increase the reproductive rate, and then we release animals back. There's a whole variety of things that we could do. The next step is to monitor that population to test the outcomes of those activities. So managing genetic diversity still requires an ongoing monitoring of that diversity. So at the population level, why would we do this monitoring? Well, if we do choose to do nothing, because we maybe assess a population as not being at great risk, we still want to check in from time to time and make sure that our predictions are borne out. So if we predict that a population will be fine without action, we probably want to check in and make sure that nothing's changed and that that's still true. On the other hand, of course, if we do some kinds of management actions, we would want to check in and make sure that those actions are indeed having the desired effect on population parameters. At the individual level, this genetic monitoring can also give us additional information about how the population is responding to our action and how it may be changing in the future. We can look to see if there are individual level variables that contribute to survival or reproduction. This information can help us to plan future actions if necessary. And we can also determine whether migration among populations is occurring. So whether or not we have increased connectivity, if we've changed something, then it may change the migration patterns of that population. So the genetic data can help us to figure that out as well. And key to all of this follow-up monitoring is to then design follow-up management that will help us to stay on track towards our goals. So this is in a context of a process you might have heard called adaptive management, where we try an action to solve a problem or achieve a goal, and then we keep an eye on the outcome and we use additional action to make sure that we stay on track if something happens that we didn't expect. What could happen? Well, a couple of examples. Sometimes solving one problem can introduce new challenges. For example, if you bring individuals into a breeding program so that you can increase the reproductive rate, now you've got to be really careful to make sure that you're preventing inbreeding, because that breeding program population is probably quite small. So inbreeding is something that can happen in a small population, as we know. So you've got to make sure that trying to solve one problem, increasing the reproductive rate, doesn't introduce a new problem. And Zoo and Aquarium Association is very good at this. They have software and they use to try and prevent inbreeding in the small population. Or similarly, if you translocate individuals to a new site, maybe translocate a small number of individuals, and only a handful of individuals end up dominating the breeding of that population for the next generation. So you may have a large number of individuals that don't contribute, And what's happened then is that fewer of your individuals have actually contributed to the establishment of that population than you thought or than you translocated. That's not uncommon. I've seen it happen in examples from mammals and birds. So definitely something to keep an eye on. Maybe future translocations are required to prevent inbreeding in that population. Also, of course, if you translocate individuals, they may disperse from the population and not stay at the site. It's definitely a question that is often asked during translocations, and genetic monitoring can help to answer that question by testing the next generation of individuals produced in that population to identify whether they perhaps show any genetic signatures of being the offspring of the translocated individuals. And finally, as I said on that previous slide, they had that big diagram with all the flow chart for genetic management. We can use the lessons learned from our previous activities to plan future ones. So that's just a very quick sort of whistle-stop tour about some of the genetic processes that apply in a conservation context, how we can measure and understand those different genetic processes, and how we can use that information to plan management strategies. So today we're going to be looking at a study system that both Kate and I are very familiar with, and many of you in the room will have come across the system before. We're looking at the Tasmanian devil, and the data that we're using is from a published study that Kate has worked on with in her lab group. So we'll talk about that in a second. But just in case there's anyone on the call who doesn't know, Tasmanian devils are a marsupial. They are the largest carnivorous marsupial. They are found only in Tasmania, down in the south of Australia. And they were previously found all over the state until 1996, when there emerged a cancer, a very unusual cancer, devil facial tumor disease, it's called. It's a contagious cancer, and we don't normally think of cancer as being contagious, so this is a very unusual disease. It's spread from one devil to another when they bite each other during fighting and mating. And unfortunately, it's very deadly, and individuals almost always die from the cancer. It has spread throughout Tasmania. It's found pretty much almost everywhere. There's a couple of pockets where it hasn't been detected, but more or less throughout most of Tasmania, and it has caused massive population declines. Now, in response to this rapid decline in the population, the Tasmanian government and the federal government joined together and formed the Save the Tasmanian Devil program and established a range of interventions to try and protect the devil. So some of those interventions have involved the establishment of a series of populations that can, to varying degrees, protect the species from extinction. So I've put this diagram here, which shows sort of a summary of the types of facilities and the types of populations that have been established. So on the right-hand side, we've got the wild, which includes a series of localities across Tasmania that have been very closely monitored over several years. And some of that data is what we'll be analysing today. And then we have a continuum of different levels of conservation intervention, all the way up to, on the left, intensive, which is your zoo-based breeding enclosures. Then we have in between that managed environmental enclosures and free-range enclosures, different size populations, different size fenced areas, and then Maria Island, which we'll talk about a little bit more in a second. So the purpose of all of these different facilities is to try and achieve the benefits of all different kinds of conservation strategy. As I said before, sometimes when you do one conservation action, it presents challenges for trying to achieve alternate conservation objectives. And the continuum that you see here on this diagram shows that if you use a range of strategies, you can achieve multiple benefits. So, for example, with the intensive population, that first benefit is that you can control the breeding, so you can minimize inbreeding. But in the wild, that there is natural mate choice. So we have that whole range of different benefits there. So this is the paper. I would like to acknowledge all the contributors to the paper, Kate, who we're very lucky to have on the call to talk us through it. We will be analysing just a subset of the data from this paper. So if you're interested to learn more about how this data is used for devil management, then please go and have a look at the paper. I believe this PDF is available to you in your resources. So yeah, the full analysis is in the paper and we'll just be looking at a small part of it. And this is where all the data have come from. So this is a map of Tasmania, and each of those coloured circles represents a locality where individuals are sampled from. Look at those sample sizes. How impressive. There's just a huge number of samples available, and that is in large part due to the massive efforts of our colleagues down in the Tassie government who have been monitoring these populations very closely for a really long time. So included the insurance population, which I've just sort of pulled out as a separate thing down here at the bottom. That's the zoo population that I mentioned before, that's held over a number of facilities across Australia, and Maria Island. Now, Maria Island is interesting because it didn't previously have devils on it, or it hasn't recently had devils on it, but devils were translocated to Maria from the insurance population as another sort of insurance for the species. So the devils on Maria Island are largely representing the insurance population. That's where they're from. Oh, and I should say that the insurance population was established from individuals from mainland Tasmania from a variety of places. The devil facial tumour disease started up in the northeast, and then the disease gradually spread across the state. And because at the time it wasn't really known how the disease was spreading or what the consequences were going to be, obviously when it was first discovered, that research had to be done. So initially, individuals were taken from ahead of the disease front. So healthy individuals that hadn't shown any signs of disease were used to establish the insurance population. However, as time went by and researchers learned a bit more about how the disease manifested, how it grew and its effects on the devil itself, they were able to implement a range of quarantine and other procedures that enabled them to bring devils into the insurance population from other parts of Tasmania as well. So there was a bigger representation of individuals from the insurance population, from the wild, or I should say, that was the plan, and that's part of the questions that Kate will be talking about with this analysis. So some of the sites that are included in this analysis, based on earlier microsatellite studies, it had been shown that there was sort of an east-west gradient in the diversity of devils. So samples for today's analysis will be taking a few western localities, a few sites from sort of the central region, and then a few sites from around on the east. So Kate, would you like to add anything about this context?
Thanks, Catherine. I think that was a great introduction to the study. So yeah, I guess just to say that we've chosen for the purpose of this workshop, the populations or the sites where we had sampled at least 20 individuals. And so this is just a subset of the data so that it's quicker to run during the workshop, but the full data is publicly available with the paper as well. And the paper's open access, so everyone should be able to access it. I guess in terms of the workshop and the topic that Catherine has so well introduced, here we have a series of often quite small populations, and the ones that we have more sampling effort for are often the sites where the Save the Tasmanian Devil program have returned to every year for monitoring, including genetic monitoring, so taking an ear biopsy. And then some of the smaller dots you see in between have either been targeted sites where they were deliberately, people deliberately went there to try and sample devils to try and fill in some of the gaps in this map. So we could look at the patterns of genetic diversity at a finer scale, or they were part of a citizen science project where members of the public actually reported devils that had been hit by cars and were roadkill, and samples were collected from those devils, which also helped to fill in some of the gaps in the map geographically for sampling. So I think that then leads into the objectives of the study, Catherine, and also of today's workshop.
So yeah, the original objectives of the study were to quantify genetic diversity of devils across their entire range, as Kate said, and then also to investigate whether genetic diversity of the insurance population is representative of the species. So as I said, there was, for very good reasons, there were some operational constraints on the way that the insurance population was established. They needed to use healthy animals, individuals with no disease. So they were initially taken ahead of the disease front towards the west, but then later individuals were introduced from elsewhere. And then individuals that were released onto Maria Island are then being used to help supplement populations on the mainland of Tasmania that may be suffering from small population challenges. So some simulation modeling has shown that these populations may be small. These small populations may be really vulnerable to losing genetic diversity and increase inbreeding. So it's really useful to now collect a wide range of high quality data and make sure that these management decisions are achieving the goals that the program has set out.
I'll just add there as well. So the insurance population has been very successful for Tasmanian devils. So it's grown a lot since it was established in 2006, to the point where it now is at that serving its purpose, where we can actually then reintroduce devils back into the wild. And so that's why this study really came at a time where it was important to see, firstly, whether the insurance population did capture the genetic diversity of the species as a whole, which is one of the goals of a captive insurance population, but also whether releasing devils back to the wild from who have originated in this insurance population, what impact that may have on genetic diversity, depending on how the genetics looks within the insurance population as well. So today's activity that Kate will lead us through some of the objectives then, first to replicate some of the analyses, which will include calculating some of the EBVs that we talked about for a subset of populations. We can have some discussion about the findings in relation to devil management, or more generally, how we can use the kinds of results that we're generating to think about and plan alternate management strategies. And then also consider the implications of sampling and study design for addressing these kinds of questions. As Kate pointed out, some of the sites that are not included today were strategically targeted to identify localities that were underrepresented in the main sampling program. And so by having a think about what we find today and what kinds of questions you might have about whether including those additional data points will provide additional data for making management plans. So I will hand over to Kate.